Title of today's lecture is Chemical Synapse. And the synapse is where nerves come together. Come together right now. Anyone? No. Aerosmith? Alright. Over me. Alright, let's see what we remember. Hopefully that wasn't a copyright violation. I think if you do a terrible job singing, they can't really hold you accountable. Alright. So, what's this round part called? Soma. What are these little dangly things?
Dendrite.
There we go. What's that guy?
Axon.
And these are the axon terminals. Let's say in the neighborhood here. Hello, neighbor. We got this guy right here doing his dendrite thing. And right now, while you're learning this information, here's what's happening. This dendrite's like, I'm gonna touch that one, but not quite touch it. It's gonna get as close as it can. Can you see the little gap? See that little tiny gap right there? See that? Learning new information, folks, is new connections happening between neurons in your brain. That's what learning actually is. But they actually don't touch. There is this little bitty gap that exists. I tried drawing this earlier today, and I laid too much ink down at that gap, and then it rolled over and it closed the gap, and I had to white it out. It's close. It's just really close. And that gap right there is a chemical synapse. Synapse is just the name of the connection or joint. Chemical is more specific about what's happening there. That is a chemical synapse. And if we take this dude down here and we take a really good close look at it, we're going to see, here's one part there, and there's another part right there. That doesn't look perfect. It's okay. Doesn't have to look perfect. This right here is the axon terminal. And if I can be a little more specific, this will be nerve A, this will be nerve B. Axon terminal of nerve what? A. And this is the dendrite of nerve B. So we're getting a really good close-up look here at what's happening there. Note that they don't touch. They're not supposed to touch, okay? If they did, you'd have all kinds of a mess of electrical activity. We need a gap to be able to control, to be able to stimulate some neurons while kind of relaxing other neurons and so on and so forth. This gap's particularly important in the brain. Okay. Because we got to be more specific and everything's got to have the word synapse all over it, so that's what we're going to have here. This big knob here thing at the top is sometimes called the synaptic knob. Synaptic knob. This space between the two right here is called the synaptic cleft. Not to be confused with clef. Clef is a music thing. It's the treble or bass. But this is like a gap. So, and sometimes they actually just call it gap too. So maybe put that here so you don't get confused if you see that.
How do you spell? Is it dendrite? DR?
D-E-N-D-R-I-T-E. Yeah, it looks like dendrite. Yeah. D-E-N-D-R-I-T-E. Dendrite. Okay. Synaptic cleft. All right. And these are cells. So this boundary here is actually a cell membrane made of phospholipids and so on and so forth. But this particular one, because it's right before the synapse, is called the presynaptic membrane. And this one that's right after it, you can probably guess what it's called. postsynaptic membrane. Postsynaptic membrane. All right. So then in here, ladies and gents, we're going to have a bunch of these little circles, courtesy of our friendly neighborhood Golgi apparatus. You remember in studying cells and biology that the Golgi is the thing that packages stuff for transport. Well, it packages them in these little ball things of basically made of membrane material, made of phospholipids here. And they're called vesicles. But because they're vesicles that deal with the synapse, guess what they're called? Synaptic vesicles, right? Those are synaptic vesicles. And they are filled with, absolutely filled with, and I'm going to draw dots in here, but in reality, if you saw these in an electron micrograph, you wouldn't be able to see these particles because they're less than microscopic. They're chemicals. You can't see chemicals in a microscope for the most part. And those little dots, go ahead and fill them all in there. Those little dudes right there are called neurotransmitter. Now, neurotransmitter is a general category for stuff, okay? It's a category of chemicals that passes through here between neurons at the synapse to communicate with other cells. So that is neurotransmitter. Neurotransmitter, I'm going to put in parentheses, something called ACH is most common. There's a number of different neurotransmitters that are out there that are in your brain that are at work right now. Things like serotonin, dopamine, histamine, GABA, things like that. But ACH is the most common. In fact, I'd like to sidebar on ACH for just a moment. So let's take it over here. Hopefully you got a little bit of room there. ACH is the most common. ACH, it's big A, big C, little h, stands for something called, I've heard it called acetylcholine, and I've heard it called acetylcholine. So acetylcholine. Acetylcholine. All right. It is found almost everywhere in the peripheral nervous system. Is also abundant in CNS. Well, why is that worth mentioning? CNS has got all kinds of other ones there, but in the peripheral, it's pretty much exclusively acetylcholine. Is made of an acetyl group and choline combined. I'm going to simplify the drawing here, so like basically and choline, and they're bound together like that. Now, it's a little bit more complex if you want to know what it looks like. It's not a very big molecule. If you can just look it up in a Google image search, just type in acetylcholine and you can see a picture of it. Okay? But them being combined is what allows them to do their job. And we're going to come back to this in just a little bit. All right, down here we're going to start listing some steps of what happens. Okay? 1, a nerve impulse called an, should have given a little space there, an action potential arrives at the synaptic knob. Now, bear in mind, these are biological wires. What I'm telling you is that this impulse, the signal, travels down the neuron and it's arriving here. So, let me try to give you the rundown of that. So what we're seeing here in step 1 is that that impulse has traveled all the way down the neuron and it has arrived here. Oh, so right here. All right.
You say one action potential?
Sorry, it's supposed to be and I tried to write the A there real close and it messes it up. Okay, nerve impulse called an action potential arrives at the synaptic knob. This triggers rapid exocytosis of synaptic vesicles. If you recall from your biology class, exocytosis is where we're taking a container full of something and sending it out of the cell in bulk. And that's what's happening here. Exocytosis. So what's going to happen? Step 1, nerve impulse arrives down here. And that causes rapid exocytosis. Like all these dudes start hauling for the border in a hurry. And when they do, their membrane fuses with the presynaptic membrane and they spill their contents out into this space here. So that neurotransmitter just gets thrown into here. Okay? All right. Now, that wouldn't matter except for this. Okay? Right after that happens, you have these little, on the postsynaptic membrane, there are these things. And if you say this, you have to say it like a boss. So what are these? These are proteins. These are protein receptors that are on the postsynaptic membrane. And ACH goes and binds to them. So it binds to them. So, and again, it doesn't have to be ACH. It can be any neurotransmitter. So, neurotransmitter floods the synaptic gap and binds to receptor proteins on the postsynaptic membrane. Slide that up. Think about this like, let's you write that down for a second. Neurotransmitter floods the synaptic gap and binds to receptor proteins on the postsynaptic membrane. This binding causes the neuron B to fire. Action potential. Okay. So that binding causes this to happen. So as soon as these things, so one more time in a nutshell, action potential arrives here, causes a rapid exocytosis of these things. That neurotransmitter floods this space, binds to here, which causes the action potential to travel on and through into the next neuron. It's these chemicals that make that happen. Now, here's the unfortunate thing about acetylcholine. It bonds so well with these proteins that it doesn't let go. That's not great. So, there is this enzyme, all right? And I want you to draw it. Enzymes are all proteins, like a boss. So here it is. Make sure you draw it exactly like I have it. That's typically what a protein looks like. Somewhat of a jumbled mess like that. I'm going to get take a little bit of creative license here and draw a little hand coming out of it. Okay, note there is no little hand in this enzyme. And in the hand I'm going to put a little pair of scissors. And what happens is this enzyme, through very complex chemical means and not with a little hand and pair of scissors, it will snip this bond between those and separates them into an acetyl group and choline. Okay, so they get split up. This enzyme's abbreviation is ACHE. I know that looks like the word ache. Okay, I'm aware of that. Capital A, capital C, capital E, lowercase h. Yeah. So that enzyme is known as acetylcholinesterase. ACHE is fine if you know that. What it does is it splits those up so that they can be removed. Okay. Fun fact, well, not fun fact, it's actually really gross and cruel now that I said the word fun, okay? Bug killers, insect sprays, the way that they work is this. They block this enzyme from doing its job. So basically, if you've ever seen a bug die from bug spray, they twitch a lot because they can't stop twitching because their nerves will not stop being stimulated. Basically, they twitch themselves to death, like in every neural junction in their body. You think, wow, that stuff's probably not good for us either. Yes, don't breathe a lot of it, okay? But it won't kill you to spray some bugs because you're big, they're little. Not that that makes it okay, I'm just saying that that's why you don't die when you spray bug spray, okay? So, yeah, so then I'm going to finish this up here. Removal of ACH by ACHE. Removal of ACH by ACHE. Splitting ACH molecules. And then last step would be reabsorbing acetyl groups and choline by presynaptic membrane. Okay, now again, not every neurotransmitter is ACH. I'm just showing you kind of how that works. So ACH is just kind of floating around in there, ready to break these things up to kind of reset this process. Okay? So enzymes, if you remember, what an enzyme actually is a catalyst. It basically makes a reaction happen. So if you want to look at it this way, acetylcholine here's the reaction, and then the enzyme ACHE breaks it down into acetyl group and choline. I believe in chemistry you call this a decomposition reaction. You take a larger molecule and you break it down into something smaller. That's basically what's happening here. So That's it in a nutshell, gang. But again, ACH is not the only neurotransmitter. There are a number of different neurotransmitters, and we'll start with that one because A, it's the most common, and B, it's a great example of this enzymatic activity. So that is chemical synapse.